This recorded session is dedicated to our Module 3 in the lecture component of Industrial Microbiology. So, we have now completed our discussion on the history and purview of industrial microbiome. We also covered important topics that will be recurring throughout the semester, such as the design and types of microbial bioreactors. We also discuss um, the process of or the workflow of isolating potentially industrially significant microbes. And we also talked about sources of these microbes in natural and artificial settings or environments. Now, we will have a review on the characteristics of microorganisms, the requirements for their growth, and we will talk about some examples of microbes as applied in different dimensions or different industries, which are relevant to the industrial scale use of microbes and bioproducts that they produce. So let's have a quick review on microbial requirements for growth. So in scope of our course include organisms that are not only prokaryotic, not only bacteria, but also eukaryotic unicellular organisms such as fungi, particularly molds and yeasts, some virus, algae, and protozoa. No? So, by definition, industrial microbiology relates very well, or in a way, you can look at it as the same with biotechnology. So, in these two disciplines, we exploit biological systems, their their the mechanics of their cells, the products that they produce, or the products they produce in relation to daily living or the daily thriving, and we look at how these products or we optimize how these products can be produced since they offer market value since they have economic importance. Okay, so all these microbes, both prokaryotic and eukaryotic unicellulars, have physical and chemical requirements for growth, and some of them we will cover in this lecture. So for the physical requirements, the first one that we always remember is temperature. So microbes have optimal temperatures that allow them to grow and reproduce, allow them to divide and increase in terms of cell number. And based on temperature, we have some types or categories of microbes. So we have psychrophiles, which have preference to low temperature, usually at freezing temperature and up to 20 degrees Celsius. Mesophiles, which grow at temperatures between 20 and 40 degrees Celsius, optimum approximately at 37. Most medically significant microbial pathogens belong in the mesophile criteria. And then we have the thermophiles, which prefer high temperatures, 40 degrees to 90 degrees Celsius. No? So we also have a special case for another group of prokaryotes, the archaeans, which are extreme microorganisms. They have optimal growth temperatures that are set at 80 degrees or above. No? And I think in the previous lectures I've provided a number of examples on how, or a number of examples of archaeans that are utilized, no? That are exploited because of the enzymes that they produce, since those enzymes are industrially significant, no? Enzymes that can withstand very high temperature, very low pH, or high osmolarity. Okay, so when we look at how, or if we look further, no, for temperature, we have this cardinal temperature curve, which is just the representation of the spectrum of temperature points or degree points that the different groups of microbes prefer. So there's a peak as represented here, okay, as represented by these areas, the peak, which is the optimal temperature. There is a minimum, as shown here, and of course there are maximum. And you can appreciate that from minimum to the optimal temperature, there is increase in the growth of microbes. There's increase in their growth rate. And down from cardinal, or from the optimal temperature, nearing the maximal temperature that they can survive, there's a decline in the growth rate. As microbes will no longer tolerate more temperature or higher temperature on the other side of their spectrum. Okay? Then we also have pH as another physical requirement. So pH, as a review, refers to the acidity or alkalinity of an environment, of a solution. And based on on pH values, on tolerated pH values, and preferred pH values, we have acidophilic microbes that grow in acidic environment, less than pH of 7. Neutrophilics, which grow in a neutral environment, pH of 7, 6.5 to 7.5. And of course, alkalophilics or alkaliphiles, which grow in basic environments, pH with higher than 7 value. No? Okay. Then we also have osmotic pressure as another physical requirement. So any change in the concentrations of solutes in an environment definitely affects microbial cells, as differences in solute concentration will drive water movement. Okay, so as a review, I'm sure this is the end time that you probably encounter these terms on tonicity. So we have hypotonic, hypertonic, and isotonic solutions. Hypotonic solutions are solutions that allow water to move into the cell. Okay, primarily the solution is of lower osmotic concentration relative to the internal fluid or the internal environment of the cell. So water will move to dilute, will move inside to dilute the concentration inside the cell. And when that happens, the cell will swell and might result to bursting or lysis. Then we have hypertonic solutions or environments. The external or the ambient has a higher osmotic concentration relative to the internal environment. Water moves outside the cell to dilute the ambient, and membranes shrink, resulting to plasmolysis. Okay. Then we have isotonic solution, meaning the environment or the solution has the same osmotic concentration as the intracellular fluid, or similar with the intracellular fluid, meaning there is no net change in water concentration or water volume. There's no net change in the um, uh, flow rate or movement rate of water molecules inside and outside the cell, or going outside and going inside. So whatever enters the cell is the same as the number of molecules that exit it. So cells in isotonic solution remain normal. So in our laboratory experimentations, we do or we make use of isotonic solutions in order to dilute our sources, our samples, diba? Before we plate them. Okay, so just a visual, set of visuals on on cells as they are present or as they exist in isotonic solution and a hypertonic solution. Okay. All right. So let's have some examples, some representative organisms that fall within the different categories of physical, the within the spectrum of physical requirements that dictate their growth, dictate microbial cell reproduction. So for halophilic organisms, some examples include Halobacterium, Dunalia, and Ectothiorhodopspira. So they require very high levels of salt, sodium chloride, about zero point 0.2 molar to grow. And then we also have, for those that are tolerant or tolerating of um, um, changes in the osmotic concentration or osmotic concentration of the ambient. We have Staphylococcus aureus and Saccharomyces species. Then within the pH bins, we have the following. So those that prefer acidic environment include Sulfolobus, Ferroplasma. Sulfolobus and Ferroplasma are microbial are prokaryotic genera that are usually associated with mining, no, with mining plants. Then for neutrophilic organisms, we have the usual Escherichia, and then some eukaryotic unicellular microbes such as Euglena and Paramecium. Then for those that prefer alkalic or alkaline environment, we have Bacillus alkalophilus and Natronobacterium. For temperature, so we have the following examples. You can see that um, the mesophilic group usually contain the either normal microflora, transient or resident ones in relation to the human body. Then for thermophilics, we have the following: Geobacillus, Styrothermophilus, and Thermus aquaticus. Then for psychrophilics, we have Bacillus acrophilus and Chlamydomonas nivalis. So these psychrophiles might be considered as potential threats in terms of food safety, as they can survive or has optimum temperature growth rates or temperature related growth rates at cold, no, or freezing temperature. And then for oxygen concentration, um, we have the following. Though for oxygen concentration, it's not really physical requirement anymore, but um, as an example or to sum up what we have so far. So you have microbes that depend on molecular oxygen in their energetic needs and carrying out their energetic activities. So we have obligate aerobes. Then we have those that do not really require oxygen but might grow better in its presence. So we have facultative anaerobes. Those that are tolerating of very small concentrations of oxygen, such as Streptococcus pyogenes. Those are aerotolerant anaerobes. We have obligate ones, obligate anaerobic organisms. So we have Clostridium, Bacteroides, and Methanobacterium. And microaerophiles, we have organisms that require oxygen level at very low percentages and usually damaged by atmospheric levels or concentrations of oxygen, such as Campylobacter, Spirillum, and Treponema. Then we have those that grow rapidly at high hydrostatic pressures, such as Chivanella and Photobacterium. So those that are categorized or identified as piezophiles. Okay. Then we have the chemical requirements for microbial growth. So chemical requirements refer to the atomic elements that comprise the different macromolecules, um, supracellular or not supra, but ultrastructural components of microbial cells. So definitely our key elements: carbon, hydrogen, nitrogen, sulfur, phosphorus, okay, and some other trace elements that can serve as cofactors. So first we have carbon, which is the structural component or backbone of organic molecules. Carbon serves as energy source, and in terms of um, um, acquiring carbon-based molecules, or acquiring carbon, so we have chemoheterotrophs and autotrophs. So chemoheterotrophs use pre-made organic compounds as their carbon source. So they break down those pre-made organic compounds. And we have autotrophic microbes that make use of carbon dioxide as their carbon source. So our photosynthetic bacteria are autotrophic. Then we have nitrogen, which is the primary element related to amino acids and proteins. So most bacteria are capable of decomposing proteins. Some of them make use of ammonium or nitrate as their nitrogen sources. A few of them can use atmospheric nitrogen for the case of nitrogen fixing bacteria, those bacteria with nitrogenase complexes, those that can express nitrogenase or nitrogenous enzymes. And they are known as nitrogen fixing bacteria. Then sulfur, which is related to some amino acids, to thiamine and biotin. So some bacteria may make use of sulfate or hydrogen sulfide as their source of sulfur atoms. For phosphorus, of course, as another chemical requirement, important for nucleotides, DNA, RNA, and definitely the energy molecule, ATP. Also important, phosphorus is important for the structural organization of membrane comprising elements, the phospholipids, no, which have phosphate groups attached to them. So phosphate can be a source of phosphorus for microbes. Then some trace elements that are called so because they are not really required in big chunks or big volumes, unlike the common ones, unlike the carbon, hydrogen, oxygen, nitrogen, or phosphorus. No, so they are only required in small amounts as they serve usually as cofactors for enzymes. Then there are other growth factors that can be obtained from the environment, such as purines, pyrimidines, vitamins, and amino acids that microbes make use of. Okay? Another important chemical requirement is oxygen. So I've talked about this a while ago. So growth in oxygen is related with microbes, with how microbes carry out metabolic processes, whether they make use of molecular oxygen as a terminal electron acceptor. No? In the environment, oxygen is easily reduced to toxic reactive species, to reactive oxygen species, ROS. And Some examples of these include radicals, the super SO radical, superoxide radical, hydrogen peroxide, and the hydroxyl ion. And then we have for microbes that make use of oxygen as their terminal electron acceptor, they express enzymes that break down these reactive oxygen species. So they don't really harm the microbes. So some examples of these protective enzymes include SOD, superoxide dismutase, peroxidases, and catalases. In the laboratory, one of the biochemical assessments you will do is the catalase test to check if the microbe you have chosen, the colony or the isolate you have chosen, is catalase producing. So, catalase production is related with aerobic microorganisms. And then for obligate or strict anaerobic microbes, they cannot tolerate oxygen, okay, because they don't have the enzymes, and they should be grown in conditions without oxygen. So in an anaerobic or anoxic setup. So here, so some definitions on the different types of microorganisms based on oxygen tolerance and need. So aerobics, those that grow in the presence of atmospheric oxygen concentration, usually 20% and above. Aerobics, obligate ones, require oxygen. Anaerobics are only growing in the absence of it. Obligate anaerobes usually are killed in the presence of oxygen. Microaerophiles require very specific concentration of oxygen, and facultative anaerobes do not require oxygen but can grow better in its presence. So they prefer oxygen but they do not really require it, meaning their energy harvesting apparatuses or mechanisms can switch between aerobic and anaerobic conditions, no, or oxygenic and anoxygenic conditions. Then we have aerotolerant anaerobes, which grow with or without oxygen. Okay, so some representation here. I believe this is an oxygen, this is thioglycolate, fluid thioglycolate. So it's in the usual setting, the oxic zone is the zone on the upper portion. So here is the oxic zone. So those that grow here require oxygen. Facultative anaerobes, you appreciate the pattern. You expect that they will concentrate heavily on the top portion of the tube, and some are well distributed on the other portions. Aerotolerance, there's no observable or demonstrable uh, aggregation of microbes on the surface. Strict anaerobes are found at the bottom of the tube, and microaerophiles do not really aggregate on the surface portion, but will require oxygen still, but at lower levels, no? At lower vertical depth. Okay. All right. Okay, so how do we grow microbes? Definitely, we use culture media. Okay, so we culture microbes so that they can grow and multiply. We can purify them for further studies. The media that we use is a nutrient source. We prepare them in relation to the requirements of our target microbes. No? And these culture media will require um, multiple things, or will have this culture media are combinations of multiple things of water, nitrogen containing compounds, energy sources such as carbohydrates, accessory growth factors, like for the case of those that require iron as a factor, as a cofactor, then you must add some source of iron like blood, no, for the case of blood agar. Then solidifying agents, agar. We may also have pH indicators to assess how the organisms react with the media, if they make the culture environment or the medium acidic or basic. Some media may also have reducing agents and selective agents that allow for other tasks such as indication of molecule breakdown or inhibition of unwarranted or untargeted microbial groups. Okay? So, when we grow microbes on culture, on solid media, they usually grow as macroscopic visible growths, colonies, which are clusters of microbes. Our assumption is each colony came from a single cell. That's the safest, most basic assumption to have all the time. And we have several techniques in accomplishing or isolating microbes using different culture media. So it can be via streak plating, spread plating, and pore plating. Okay? So based on consistency, there are different types of culture media: solid, liquid, which we usually call broth preparation, a semi-solid medium, a hybrid between solid and liquid types. And based on the constituents, we have either a simple medium, a complex medium, a synthetic or chemically defined medium, and some special media which allows us to carry out special tasks like enrichment, selection, differentiation, and if in the case of other fields like clinical microbiology, some special media are designed as transport media. And then based on oxygen requirement, our media could be aerobic or anaerobic. Okay. Some examples, more about solid media. So, solidifying agent of solid media vary. There is agar and gelatin as examples of solidifying media. Then, chemically defined media are those that are said to be possessing or including exact chemical composition of their ingredients. Complex media are those with nutrient ingredients with exact chemical composition that are not known, like nutrient agar or nutrient broth. Those are two examples of chemically complex media. Then we also have enriched media as an example of solid types of medium. We have those that the enriched media group, which we use to support the growth of uh, fastidious microbes. So microbes that have special requirements. No. So some examples of enriched media include blood agar, chocolate agar for the growth of Diplococcus pneumoniae, Loeffler serum for the growth of Carine bacterium, and Dorset's egg medium for the growth of Mycobacteria. Then we have enrichment broth, which is essentially just the same with an enriched solid media, but this one is of course in liquid form. No? And if we want to increase the number of cells that are present in the culture. So they favor heavy growth of certain classes of microbes. So for example, uh, tetrathionate broth for the growth of foodbornes such as Salmonella and Shigella. No, so tetrathionate broth is specially designed as an enrichment medium for producing or increasing the number of Salmonella Shigella cells. Then we have selective media that contain substances that inhibit the growth of some microbes while allowing the growth of the targeted ones. Some examples of inhibitors include antibiotics, dyes, chemicals, and in some cases, we alter the overall pH of the culture medium. So, some examples here we have Lowenstein Jensen for the growth of M. tuberculosis, which will contain penicillin and nalidixic acid, inhibiting gram positives and gram negatives. No? Since you know that Mycobacterium as a genus, as a unique characteristic in terms of its cell wall. No? This special selective medium can be used to grow it. Then we have saburod dextrose agar, which contain chloramphenicol, expected to inhibit the growth of gram positives and gram negatives, but allow the growth of fungal species. Then for the growth of Pseudomonas, we have citrimide agar, containing 0.1% citrimide that inhibits the microbes that inhibits the microbes other than Pseudomonas aeruginosa. Now we also have differential or indicator media, which are media that allow distinguishing the distinguishing between types of microbes from one another. No, so the rationale is there's an indicator that will react differently to different microbes and will produce morphologically distinct colonies. So here is an example of two differential or indicator media. So I have litmus milk and urea agar. Okay. I think I may have to cut the recording so it will be tolerable on your end. So this will be part one.